Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions from the chat window. Your questions in the chat window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, David Cetlin from MockV Solutions and John Lee from Thermo Fisher Scientific.
Thank you, Leah, and good morning to everyone. My name is David Cetlin, and I'm the founder CEO of MockV Solutions. I'll be taking us through the MockV portion of this webinar, and to my virtual right is John Lee. John?
Hi, everybody. My name is John Lee, and I'm a senior scientist in the bioproduction business of Thermo Fisher Scientific, focusing on downstream applications. And today I'll be talking about monoclonal antibody, aggregate, and viral clearance case studies using the POROS HIC resins.
Thank you, John. So the way that we've organized this presentation for you is first a brief introduction to MockV Solutions, then an overview on viral clearance and the issues that it presents to bioprocess development. I will then introduce the concept of MockV's technology in the form of our MVM-MVP kit, and will then describe a study involving the use of our MVP kit in exploring ion exchange conditions in high throughput screening mode. I will then turn the mic over to John, who will introduce Thermo Fisher and their lineup of chromatography products. He will then take us through a viral clearance study which he conducted involving several POROS HIC resins. So MockV Solutions, we are a privately held tools company headquartered in Maryland. Our mission is to develop tools that lower the cost and complexities associated with viral clearance, and we'll be going into some detail in the background of this presentation. And we commercialize a series of research kits based on these non-infectious mock virus particles that we've introduced to the industry. We call them mock virus particles, MVPs for short, and we have a prototype kit available that mimics a common mammalian virus used for viral clearance spiking studies, which is MVM. And I'll be going into detail on that later in the presentation. We have that kit available for purchase, and we are performing a series of proof of concept studies with industry collaborators. So as most of you are well aware, bioprocess development is a time-consuming and resource-intensive endeavor. Many years go into the development, characterization, optimization, validation, and finally tech transfer of a downstream process. Along the way, commercial kits are readily available and used. They provide critical data on impurity removal. So for instance, Thermo Fisher has a lineup of impurity kits which really do a great job. So Protein Seq is used for measuring host cell protein removal. And similarly, ResDNA-seq can help customers for measuring the reduction of host cell DNA. The data amassed from these types of kits really form the backbone of much of the decision-making throughout the evolution of a downstream purification process. But as we can see from this slide, one impurity that's lacking from this toolbox that we have as an industry is any kit that can provide information on viral clearance. The reasons for this are fairly obvious. It's because viral clearance requires the propagation and quantification of live mammalian viruses. The environmental and logistical challenges require specialized labs and personnel, and because of this, most companies outsource viral clearance studies to contract research organizations. Because of the complexities involved in propagating virus, these studies are often pretty expensive. And these high costs unfortunately present a major hurdle to data accumulation during the early years of process development and characterization. In fact, it's not atypical for a company to delay or kick the can down the road when it comes to viral clearance until pretty late in the game, process validation, or just prior to BLA filing. And this presents some obvious risks. It's a bad time to learn if there's any particular step in the process that's underperforming. And that is a pretty severe risk, especially when there's a BLA on the line. So MockV was pretty much founded to alleviate this risk. We came up with the concept of using a mock virus particle to replace the live viruses used in typical spiking studies. And I'll go into detail on what exactly a mock virus particle is, but essentially it's a non-infectious surrogate marker for these commonly used models. And we are putting this together into a usable kit, a user-friendly kit, which includes the spiking solution of these mock virus particles and the quantification reagents required to conduct your own viral clearance analysis. So the way that a kit would work is essentially the same as what would happen at a CRO. You would spike these non-infectious particles into your load, process that spiked load through your chromatography, filtration step, what have you, and then quantify the remaining particles in the pool, and from there deduce your log reduction value. So there's many places during the lifetime of a biopharmaceutical where this concept could be applied. The most obvious are here in the middle, 3 and 4. This is basically process development, process characterization. Imagine what you could do if you had viral clearance as an output during a DOE. So that's pretty much the sweet spot of use. Early on, you can use this type of kit to screen new products that come to market, new novel techniques like continuous processing, and 2 and 5 pretty much show you periods of time right before regulatory filings where it would be good to have some insurance policy, some data leading up to those very important studies. So this kit provides opportunities to provide that data on the cheap prior to the expensive studies that you'll have to inevitably do. The value proposition is pretty straightforward for the utility of these kits. Currently, viral clearance studies at CROs are expensive, and they do take weeks of planning and execution. In terms of surrogates, bacteriophage have been used for over a decade. They do a great job at mimicking the size and physical properties of these mammalian viruses, but they're really not meant at all to mimic the surface properties, and we'll be going into some detail on that later. Also, they're not as readily available. Your lab would have to be pretty proficient at propagating bacteriophage and measuring, quantifying the amount in solution. So now I'll go into some detail on what are these mock virus particles that we have. So as I mentioned before, the first particle that we went after is meant to be a surrogate for MVM. MVM is a small non-envelope parvovirus that's commonly used as a model for viral clearance validation studies. So what we did to make our mock virus particle is we borrowed the major capsid protein of the live virus and expressed that in a recombinant system. The particles that form, therefore, are made of the same material, the same VP2 capsid protein as live MVM. And on the right, you can see a TEM image of our particles. So the first study we conducted, the first collaboration, was along with Kurt Brorson's lab at the FDA. And we asked the question, okay, so how do our particles mimic the physicochemical properties of the live viruses that we say they represent? So we provided his lab with our non-infectious particle along with live MVM, and we also used PP7 bacteriophage as another test object. And we looked at the size of the 3 particles through size exclusion multi-angle light scattering and through TEM. And we were able to measure the surface charge and hydrophobicity of these non-denatured particles through a couple assays that had been developed and used at the FDA. So the first set of data from the study is the side-by-side TEM results of the two particles. You can see morphologically the two are very similar size, similar spherical shape. So that was our first data point. And then on this slide, it's a high-level summary of all the data points. We have first the multi-angle light scattering results for the live MVM shown here in this column, our non-infectious particles here, and then finally PP7 bacteriophage here. So what you see with multi-angle light scattering and TEM is that essentially all particle types are similar, and that again is not surprising. PP7 has been used for quite a while as a good size-based surrogate to MVM. Where we really see a benefit of using our MVP particles as opposed to the PP7 is in these surface elements. So the surface charge, the PI that's calculated through chromatofocusing, and also the hydrophobicity. And so the MVM, not surprisingly, does a great job of, or MVP rather, does a great job of matching up with the MVM as opposed to the PP7. So this set of data here, and if anyone wants to follow up with me later, I'll make the manuscript available. It really set the stage for the follow-up work and gave us an indication that indeed these particles could be used to perhaps accurately predict the removal of MVM based on physicochemical techniques such as chromatography and filtration. So next slide, slight transition here. So we have these particles, but how do you quantify them? They're non-infectious and they do not contain nucleic acid. So a straight-up qPCR is sort of out of the question. So at first we developed an ELISA assay, simple ELISA, but the sensitivity was pretty limiting. So what we did is we came back and developed an immuno qPCR assay, and I'll briefly describe how this works. We have 96-well microtiter plates coated with an anti-MVP antibody. When sample is applied that contains the particle, it will bind. After a washout step, we then add a conjugated version of that capture antibody. The conjugated product is actually a piece of DNA, a template strain of DNA. So now we've externally recruited the DNA to any bound MVP. The DNA serves as a template for running qPCR. So on the right-hand side, you can see a typical standard curve going from right to left, the dilution series of the MVP, and the CT values, which is the output in terms of qPCR. And what we see is that over basically a 4 log range from 1e to the 10 particles down to at least 1e to the 6, we see a good linear response in terms of the concentration of particles. So this is the assay that we have, that we're actually beta testing and will be commercially available pretty soon. So finally, the last bit from me will be a proof of concept study that we actually conducted along with the NIH. This is specifically with the Vaccine Research Center. The purpose of the study is to utilize these MVPs to screen the performance of ion exchange chromatography resins from several vendors across a range of different pHs and conductivities. And this was done in high throughput screening mode. So briefly, what we did was robocolumns of each of these vendors' resins were equilibrated with low salt, low ionic strength at different pHs for anion exchange, slightly neutral, and then slightly acidic for cation exchange resins. So spike load, load was the vaccine center's one of their candidates. Spike load was created by spiking our MVP to a concentration of 1e to the 11 particles per mil in these adjusted loads. So after adding these to the robocolumns, the plates were mixed, centrifuged. We collected the unbound flow-through from the centrifugation and then hit the robocolumns with a series of increasing ionic strength concentrations. After the addition of each, we mixed and centrifuged, collected the samples, and then at the end, we analyzed all the samples collected for MVP concentration, and from there, we were able to determine LRV. So the next slide is quite busy, but it really does a good job through the coloration of highlighting what's going on here. So on the top here, we have it grouped by what fraction we're discussing. So for example, the load, the flow through right through the column. This is, by the way, the data set for anion exchange. And then going from left to right is the increasing salt concentrations which we added to the robocolumns. And what you find is that across all the resins we explored, the flow-through collections were excellent at removing the MVP that was introduced. And of course, this is not surprising. Anion exchange resins are positively charged. Our particle is negatively charged at neutral pH. So the resins were able to retain our particle while allowing the more acidic vaccine to migrate right through and be captured. So this operated pretty much according to how we expected. Then what got interesting was when we started to add these ionic strength conditions to the column and eluting, we saw a real differentiation among the resins and among different pHs. One of the high performers here was the POROS 50 PI. Across all the different pHs tested, we received good retention of the MVP particle on the resin all the way up till about 300, 350 millimolar salt, which is quite surprising. So the conclusions from this study were the porous anion exchange resins demonstrated a robust MVP clearance ability across the flow-through operations, which is generally what you would run with if you had a monoclonal antibody process. And the 50 PI resin demonstrated exceedingly good retention abilities of the MVP at high salt concentrations. On the flip side, we have here data from the cation exchange portion of the study. Cation exchange, of course, is negatively charged. At a lower pH, the MVP has a net positive charge. So that, what we see here across the resins is a general retention of the particle at the low pH, so 5.5 versus 6.5. And then as the pH increases and the surface charge of the particle shifts, we see a worsening in the ability of these resins to retain. However, the POROS XS and 50HS showed relatively good removal at either of these pHs relative to the other resins. So in conclusion, we successfully screened both anion exchange and cation exchange resins, and the data that we were able to collect through this economical and BSL-1 compatible surrogate led the NIH pretty quickly to some knowledge-based decisions to select resins for further development and potentially for scale-up. And of course, the porous resins showed a superior performance in both of those data sets compared to the other resins. And with that, I'll turn the mic over to John.
Okay, thank you, David. So as David mentioned, now I'd like to turn your attention to a couple of mAb polished case studies performed using POROS HIC, and I'll go into some of David's technology using the MockV particles as well. So just a little brief introduction. Thermo Fisher Scientific's bioproduction business, which supports developers and manufacturers of biological-based therapeutics and vaccines. And we do this with a portfolio of premium solutions focused on upstream cell culture, downstream purification, pharma analytics, as well as a suite of single-use solutions spanning across the biologics workflow. For downstream purification in particular, we have two well-established chromatography platforms, the first being the Capture Select Affinity platform based off of llama antibody immunoaffinity ligands, and the second being the porous resin platform based off of polystyrene divinobenzene based beads coupled with diverse chemistries and ion exchange, hydrophobic interaction, and or other custom ligands. So this highlighted here is a standard process, a standard three-step antibody downstream process where clarified cell culture typically undergoes first an affinity product capture step, and for affinity, Thermo Fisher has both protein A-based solutions as well as a suite of capture select affinity resins that really confer orthogonal selectivity to the protein and FC interaction, and so these are very useful for capturing FC mutated or other multi-specific antibody formats, for example. Post-affinity capture, the antibody will typically undergo multiple polish steps to remove product and process-related impurities. And it's been well recognized, at least in the mAb field, that anion exchange operated in a flow-through mode is a major workhorse for impurity clearance, such as viruses, residual DNA, and HCP. For AEX, as David alluded to earlier, Thermo Fisher has several offerings, POROS HQ and XQ strong anion exchangers, as well as POROS PI and D, both weak anion exchangers, and all of them have been implemented extensively in the flow-through mode for both clinical and commercial antibody processes. Now, additional steps for polishing are typically mAb and process dependent, and really we want orthogonal chemistry to AEX and can be operated either in bind elute mode or flow-through mode to really target those difficult impurities such as product aggregation. For example, for the second polish step, we have cation exchangers like XS and HS for polish that can be run in both bind elute and flow-through modes. But for today's talk, I will mainly focus on the porous hydrophobic interaction chromatography resins for this second mAb polish application. So our HIC resins, which were launched fairly recently, a couple of years ago, are the POROS Ethyl, POROS Benzyl, and POROS Benzyl Ultra, which covers a wide hydrophobicity range, again, to accommodate the purification needs of a wide range of biological molecules. And Benzyl Ultra here is the most hydrophobic out of the three resins. In fact, it's probably one of the most hydrophobic resins on the market right now. And in designing these resins, we really went back to the drawing board and re-engineered the base bead, the pore mode, as well as the ligand chemistries to really provide a truly differentiated product line compared to other existing HIC resins on the market. And some of the key differentiators of these resins are the highly unique selectivity, again, combined with high resolution, as well as best-in-class capacity under lower salt operating conditions. And we really also pretty paid attention to the manufacturing of these resins with a tight ligand density specification to really control that hydrophobic character. And that basically means a more robust process in your downstream purification. And since our launch, we've really seen tremendous uptake and scale up of these porous HIC resins. And in this talk, I really wanted to showcase a couple of process development case studies on how these resins can be used in mAb purification. So the mAbs I'll be talking about today are mAb-A and mAb-B. These are two clinical stage mAbs that are quite challenging to work with from a downstream perspective. Both these mAbs had a standard protein A capture step followed by, again, anion exchange flow through polish. However, post-anion exchange, there was still significant aggregation remaining, and again, of course, required additional polishing. So for mAb-A, the second polish step was set to mixed mode, operating in bind elute mode, and the scale-up process resulted basically in complete clearance of aggregation to below 1% with a 90% monomer recovery, and the process run at 25 grams per liter resin loading at a 6-minute residence time. For mAb-B, specifically, there was a particularly difficult to remove aggregate species called H2L3 in addition to the monomer and dimer species. So H2L3 was essentially one light chain more than a monomer. And after a lot of process development, the clinically scaled-up process for this aggregate clearance step was cation exchange in the bind elute mode, and which also resulted in clearance of the aggregates and the H2L3 to below 2% in the product pool. But at the same time, there needed to be a low yield of around 70% monomer recovery due to sort of the aggressive elution pooling criteria for this particular process. Again, the low density for this process for mAb-B is 40 grams per liter resin loading, again at a 6-minute residence time. So as an alternative strategy to these clinically developed polish steps, we used porous HIC resins and design processes for both mAb-A and mAb-B that had improved productivity, complete aggregate removal, as well as orthogonal HCP and viral clearance. So first for mAb-A, we performed a high throughput screening study to really map out the aggregate partitioning as a function of resin type, salt type, as well as salt concentration. And our high-throughput study really revealed regions of highest selectivity for mAb aggregates, and these are highlighted by the orange boxes in the table. So the higher the selectivity number here, the bluer it is, the better. For practical manufacturing considerations, like trying to operate at lower salt concentrations or avoiding halides and such, we just first decided to optimize a bind and elute process using sodium citrate as the salt type on porous benzyl. So to begin optimization of mAb, so the bind and elute process on porous benzyl, we first determined the dynamic binding capacity at 1% breakthrough as a function of different residence times. And here we observed that beyond 5 minutes residence time, the DBC basically approaches that of the static capacity, and that's represented by the dotted red line at around 46 grams per liter resin. And because here we wanted a faster and yet robust loading process for mAb, we set the loading density to around 32 mg per mil at a 2-minute residence time, which if you look at this is basically 90% of 1% maximal breakthrough capacity. So next we focused on developing the elution step, and we knew based on our screening studies that sodium citrate at around 200 millimolar salt concentration showed the highest monomer selectivity and elution behavior. So based on that knowledge, we optimized elution around this salt concentration, as well as the pooling criteria and residence time. So as the optimized run on the slide will depict, we used a 0.8 by 10 centimeter pipette column format, and our final elution condition was set at 265 millimolar sodium citrate with a 4 CV collection criteria and a 2-minute residence time. So using this elution criteria combined with our loading strategy, it resulted in a 99% monomer purity in the final product pool, compared to a starting low monomer purity of 88%, so removed about 12% aggregates, as well as the monomer recovery for this step was greater than 99%. So the next slide here, I'm showing the clinical process for mAb that was optimized previously, compared to our HIC bind and elute process on porous benzyl. And our process, as you can see, resulted in a higher load capacity, significantly lower residence time, so 6 minutes versus 2 minutes, with a 10% higher monomer recovery. And we do this all the while maintaining aggregate clearance performance. And the overall productivity of the process was increased approximately 4-fold while hitting those CQAs. And also note here that the ATP was also reduced from 120 ppm to 12 ppm, so offering that orthogonal host cell protein clearance to anion exchange. So next we looked at this particular process, the benzyl bind and elute process for mAb, in terms of its ability to clear MVM and XMELV in a viral clearance study. Basically, we saw complete XMELV clearance and also a partial MVM clearance in the product pool. And interestingly here enough, water and the 1 molar arginine strips was able to strip XMELV from porous benzyl. So in addition to optimizing porous benzyl in the bind and elute mode for mAb, our high throughput study also indicated the possibility of running benzyl ultra in the flow-through mode, and basically also for mAb aggregate polishing. And this can be seen by the highly selective monomer aggregate partitioning and low to no salt conditions here, again highlighted by the orange box. So again, for mAb on benzyl ultra, we optimized a flow-through process under essentially no salt. So we looked at residence time, loading density, as well as different pHs, and we showed robust aggregate clearance across different pH ranges. Data is not shown here, but finally we operated the final optimized condition at pH 7 at 2 millisieverns conductivity, so really low conductivity. And that basically allowed what we think is a straight-through purification from anion exchange to HIC without any additional buffer conditioning, also the possibility of eliminating intermediate pooling. So this run on the slide represents an optimized run with loading density of greater than 100 grams per liter resin at under 2-minute residence times. And under these conditions, because of the high selectivity for aggregates, we also show complete removal of the mAb aggregates in the product flow-through pool with a near complete monomer recovery. So again, I'm sure this is just an amalgamation. I'm showing a comparison of the clinical mixed mole process for mAb compared with the two POROS HIC processes. And clearly all process performance metrics have been improved, resulting in higher productivity. And this basically means higher loading, lower residence times, better recovery, smaller pool volumes. And in particular, if we turn to, by turning mAb's clinical process into the Benzyl Ultra flow-through process, this has the added benefit of an integrated AEX HIC flow-through process design, process train with no additional conductivity or pH adjustments and intermediate hold-up. And also, if you look at here, the filter process was also able to orthogonally remove extra HCPs, similar to the bind-elute process. And the productivity gains are 4-fold for the bind-elute process and 13-fold for the Benzyl Ultra flow-through process. So next, we also wanted to show viral clearance data for that Benzyl Ultra flow-through process. Again, we showed very efficient clearance of XMELV from the flow-through pool. However, we did not see significant MVM clearance under the same conditions tested. And interestingly enough, for Benzyl Ultra, water is not able to strip XMELV, which really points to the strong hydrophobic nature of that interaction, and that perhaps more optimization here of the strip buffer is required. By contrast, if you recall, for the mAb bind-elute process on benzyl, XMELV was eluted by water. So next, we wanted to better understand MVM binding and clearance to porous HIC resins, and we took advantage of David's MockV MVP technology, MVP particles, which again mimics live MVM. And we perform high-throughput screening of mock virus binding under increasing concentrations of sodium citrate for the 3 porous HIC resins. And here, as the graph shows, we saw less than half a log binding clearance of the MVP on all 3 HIC resins, even under the highest salt concentration tested here, which was 600 millimolar citrate for mAb. And overall, these observations point to the hydrophilicity of MVP and therefore, by extension, MVM, that is consistent with existing literature. And this also is consistent with what we saw in live viral clearance studies, where there was minimal binding clearance of MVM for Benzyl Ultra flow-through process. And similarly for the Benzyl Ultra bind-elute process, we also did not see MVM binding during the load of mAb at 600 millimolar citrate. And so taking advantage of this, perhaps adding a high salt chase in the bind-elute process would have improved MVM RLV in the elution pool. So here I'm just summarizing the data. It's a side-by-side comparison of the live viral clearance study. And so MVM clearance as either a single spike, cocktail spike, or the MVP study done at BSL-1, just on the benchtop. You can see for both mAb bind-elute and flow-through processes in green and orange, the MVP clearance is very comparable to the MVM clearance within a half a log range. And this really supports further use of the MockV particle kits as process development tools to really predict and map out these process conditions. So the next couple of slides, I'll switch gears a little bit to talk about another mAb, mAb-B. Again, this mAb is unusually difficult with an aggregate species called H2L3, which is basically 1 light chain more than a monomeric product. Both these mAbs have around 10% aggregates after an exchange polish, so very difficult to work with. And the clinically optimized polish step was the cation exchange bind-elute and operated at 6-minute residence time with a 40 grams per liter resin loading. Again, they're accepting really high yield losses, mono-recovery losses, because they're really cutting off that elution pool quite aggressively at the tail end. So we designed, as an alternative to CEX, a porous Benzyl Ultra bind-elute process. And I'm just showing the metrics side by side here compared to the clinical cation exchange process. We managed to increase the yield by 20%. And we also basically show the similar aggregate clearance in removing the H2L3 species. And at the same time, we're running it at a lower residence time. At the same time, we also did a quick bind and elute process for mAb on porous ethyl, strictly for looking at viral clearance without really optimizing further the aggregate clearance on this particular resin. So first we took a look at the viral clearance on the Benzyl Ultra process using mAb. And here you can see that we bound the mAb at 700 millimolar citrate. Looking at the flow-through pull fraction, and it's interesting that we see both XMELV and MVM binding to Benzyl Ultra under the salt condition. And as expected, no salt buffer selectively eluted both monomer as well as MVM. Therefore, we really saw minimal MVM clearance, and it's consistent with our previous MVP high throughput study. But we did see, also consistent with our previous live viral clearance studies, that we did have complete XMELV clearance in the product pool. And note that water here, because it was Benzyl Ultra, again, was not sufficient to elute XMELV off of the Benzyl Ultra, so that basically that means more optimization of the strip buffer is needed. Finally, like I said, we also looked at viral clearance on porous ethyl, but this was an unoptimized mAb bind elute process. Again, we bound the mAb at 700 millimolar citrate, and we saw binding of both XMELV and MVM to porous ethyl, albeit to a lesser extent for MVM, likely due to a relatively less hydrophobic resin being tested here. In the mAb elution pool, again, we saw complete XMELV clearance and modest MVM clearance. And for ethyl, in contrast to Benzyl Ultra in the previous slide, simply a no-salt tris buffer here was sufficient to elute both XMELV and MVM. And also note that this relatively easy viral strip behavior was also observed for mAb bind elute process on porous benzyl. So basically, benzyl being more hydrophobic than ethyl, but less hydrophobic than Benzyl Ultra. So lastly, I'd like to conclude this part of the webinar. We've shown that POROS HIC resins are highly efficient at aggregate removal while offering orthogonal HTP and viral clearance capability. And for all the viral clearance studies we've done, we've shown complete XMELV and partial MVM viral clearance for both mAb-A and mAb-B case studies. And we show that XMELV binds really strongly to POROS HIC, and very weakly, and MVM binds very weakly to POROS HIC resins. Also, MockV MVP technology at BSL-1 really closely paralleled MVM clearance at BSL-2 in all our studies. And we can use the MockV MVP technology in high throughput format to really predict and design MVM clearance behavior through the POROS HIC resins. So just to conclude here, and then on key takeaways for the webinar, for Thermo Fisher, we have best-in-class ion exchange and HIC resins on the POROS platform, and I think we've shown here the ability of these resins in the context of viral clearance as well as impurity removal. For POROS HIC, which was the focus of this particular webinar, we've demonstrated superior aggregate clearance and as well as impurity clearance, and finally viral clearance as well. And I think really that stems from the uniquely designed selectivity of these resins for these impurities. And I think HIC in general would be an excellent and flexible downstream tool in the toolbox for all kinds of biotherapeutic capture and polishing in general. And specifically for mAbs, I think it's also a platformable polish step in your downstream process.
And on the MockV side, we were able to demonstrate that we've developed a BSL-1 compatible surrogate. We've demonstrated that that surrogate is physicochemically resembling the live MVM virus used as a model for spiking studies. And we showed some data that highlighted the utility of our MVP for predicting MVM in anion exchange, cation exchange, and hydrophobic interaction chromatography, both in high throughput and traditional screening studies. So finally, I'd like to acknowledge, jumping around the slides there. Well, I can't acknowledge myself, but Dale Dembro, he's the head of CMC at MockV Solutions. And I'd also like to acknowledge two people that are not on this deck here from the NIH. That's Dan Goetsky and also Krishna Gula. And I'll actually update this slide before it's disseminated, containing their names as well.
Yeah, and on our side, Thermo Fisher, I'd like to really acknowledge your Fins Lancaster Laboratories for performing the live viral clearance studies with us, and also Immunogen for graciously giving us the mAb-A and mAb-B to work with as case study examples for these POROS HIC resins. Okay. Thank you, and we'll take questions.
Okay, great. Thanks, David and John. So the first question, it looks like, is for David. The first question is, are the MVP particles prepared using a baculovirus system? And if so, how do you ensure the absence of infectious baculovirus from your MVP preparation?
Yeah, great question. So up until this point, we've been using TEM as the way to inspect the impurity elements in the preps that we have. So through TEM, you can pick up the presence of baculovirus or even debris. However, we acknowledge that there's probably more sensitive and accurate ways of determining or validating that indeed our preps are free of baculovirus. So we are going to be transitioning from that approach to a more kit-based approach. There are several commercially available kits on the market that can be used to detect baculovirus. So that is something that we will begin to implement.
Okay, and is an MVP a good surrogate marker for a low pH hold inactivation step as well?
Yeah, that's another good question. So we've actually, we do have a couple customers who have purchased the prototype kit for that purpose. I'm not privy to all the data that they've collected, but I did run an experiment where I spiked a protein A pool with the MVP, dropped the pH to around 3.2 for a half hour, and then titrated back. And I looked at the signals both before and after the low pH adjustment. And what I did notice was about a 1 log removal of the particle. I'm not in a great position to say if that's comparable to what you would see compared to MVM, but that's the data point that I have to work on to answer that question.
Okay, and I think this question is for John. Where and how would HIC be implemented in a mAb downstream purification platform?
Okay, so again, I think in my conclusion I mentioned that HIC is quite a flexible unit operation in the downstream platform, and really we can implement it anywhere, post-protein A, like post the viral hold and before AEX flow-through or after AEX flow-through. And again, it can be done both in bind elute or flow-through format, although for recent trends in process intensification and such, probably running a flow-through mode is a better option. And I think I've shown this with mAb-A's process development as well. You see that in the flow-through mode, you get about 14-fold increase in productivity compared to the bind elute HIC process, which was only 4-fold. But it really depends on the goals of your process, I guess. If you really wanted more MVM clearance, for example, or somehow your ACP is dependent on a bind elute process, then you would do that. But I think the HIC in general is really good for aggregate polish and removing those impurities that is basically having orthogonal chemistry to the IEX.
Okay. I think this question is also for John. Can POROS HIC be used to polish bispecific antibodies?
Yes. So HIC is really designed to be used in a variety of different biotherapeutic polishing and or capture. So we do have customers using these POROS HIC resins for bispecific antibody polishing and to remove both aggregates, low molecular weight and high molecular weight species. So yes, that is definitely possible, and our customers are using these resins for that purpose.
Okay, so this may be a question for both of you. Have either your products been used for cell therapy endpoints such as removal of lenti or retroviruses from CAR T culture?
So I guess I'll start off. We don't currently have a commercially available kit for retro. However, we are in the process of developing that actually through an FDA grant. So hopefully by early 2020, we should have that available. And once it is, it could be used for mAb processes, CAR T processes, any process that needs to demonstrate the removal of a retro particle.
Yeah, and to follow up on David's point, yeah, certainly we have seen examples of porous resins being used in gene and cell therapy. For example, specifically in AAV, our anion exchangers, HQ, and some other resins in the anion exchange portfolio have been used to, separate and polish AAV. And for HIC, it's also a possibility. Really depends on stability of for example, the AAV particle, and there have been some examples of polishing using HIC. Of course, you need really high concentrations to bind the particle to the HIC. And again, I think I've shown in this webinar that, for example, MVM is actually a very hydrophilic virus particle, and by extension, since AAV also is part of a virus family, we expect the hydrophobicity ranges to be similarly less hydrophobic. So really depends, I think, on the process you're trying to design here.
Okay, so thank you everyone for your questions. I think we just have time for one more question. If we didn't get to your question, they will be passed to our speakers and they'll follow up with you individually. So the last question I think is for David. And how did you verify that the MVP particles are empty?
Oh, that's a good question. So theoretically, these particles during the recombinant expression, they could pick up some host cell DNA as they assemble. So it is theoretically possible that they're not quote unquote empty, completely empty. But I guess what's more important there is there's no genetic element of live MVM present during this whole entire process, except for the VP2 capsid protein, of course. But that in and of itself, even if it were to make it into the particle, would not confer any sort of infectivity. And in fact, we did demonstrate through a study with TechCell that our MVP particles are non-infectious based on MVM's infectivity assay. So just to summarize that, theoretically there could be, let's say, host cell DNA, like SF9 DNA packaged into the particles, but there's no possibility that these particles are containing infectious conferring MVM nucleic acid.
Okay, well, thank you very much, David, and thank you, John.
Okay, thank you.
Thank you, guys. Thank you all for taking the time today and for your interest.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.